Hi everyone, today we are going to talk about the concept of enzymes, which, as I said in my last video, my next video, let's say this video is going to be my second video related to the concept of enzymes, which in this moment we are going to talk about, which today we are going to talk about the optimum temperature, the optimum pH, and also the concept of denaturation. So in this moment, let's talk about the optimum temperature. The optimum temperature is a certain temperature which that enzyme can work at its best, or let's say at its greatest and its fastest rates. The optimum temperature of enzymes are specific. That means they are different for each enzyme. For example, the optimum temperature of the enzymes in our body are 37 degrees. And also in plants, it is going to be approximately equivalent to 28 to 30 degrees. There is a tip here that if the temperature rises from an enzyme's optimum temperature, it denatures, which means that they cannot catalyze any substrate anymore since the shape of their active site has been changed over time. That this concept, or let's say this process, is completely irreversible, and those enzymes that they had been denatured before, they cannot regain the last shape anymore. That means they are done, and they cannot catalyze any substrate anymore. And also there is another tip here that if the temperature goes below the enzyme's optimum temperature, it does not denature. Don't forget this. If it goes below the enzyme's optimum temperature, it does not denature. But the enzyme cannot work and catalyze its substrate in its greatest and its fastest rates. Or let's say, in other words, in a summary, they will catalyze slowly. And as you can see also in the picture, that before the process of denaturation happens, this means before the temperature rises from the enzyme's optimum temperature, the active site is not going to change, and the lock and key hypothesis is going to remain the same. And also the enzyme substrate complex can happen often. But after the process of denaturation happens, since the shape of that certain enzyme's active site is going to change and it is going to be flexible, so those enzymes, they cannot, or let's say they are not capable enough in order to fit in those active sites perfectly, let's say. So now let's talk about the optimum pH. Each enzyme has a specific pH number, which would determine their own chemical properties. Similar to the optimum temperature, there is also an optimum pH for each enzyme, which enzymes can work at their best, or let's say, and their greatest rates in that certain point, similar to the optimum temperature. For example, the optimum pH of lipase in the pancreas being produced by the pancreatic juice is 8 for amylase, and also by pancreas by the pancreatic juice is 6.7 to 7, and also for pepsin which in stomach, which is going to be 1.5 to 1.6. What this means that it's really acidic. And there is also a tip here that if the pH number of enzymes change and get far away from the optimum pH of each enzyme's optimum pH, they will denature, they will become denatured, which the shape of their active site will change, similar to the optimum pH. But there is a difference here, that if the pH number goes down or up, it rises or decreases, no difference. It denatures, but exactly opposite, exactly different, exactly something else for the concept of temperature, for the optimum temperature, which in this case, when the temperature rises from that enzyme's optimum temperature, it denatures. But if it goes below that temperature, in that case goes below the optimum temperature of that certain, or let's say that specific enzyme, it doesn't denature. Only they will catalyze more slowly. When the pH value, or let's say in other words, or let's say in a summary, when the pH value deviates from the ideal conditions, the activity slows down and then stops. That means the activity level is going to come down as much as possible, which is going to reach the number of 0. So the activity level will be 0 and stops. So that means those enzymes cannot catalyze anymore. Since the pH scale, or let's say the pH number of those enzymes has changed that much, that means the difference between the pH and the optimum pH of that enzyme is that much huge, or it is really far, which the activity is going to stop, which causes the denaturation to happen. So as you can see, this is the pH and the temperature graph line for an enzyme. So in this case, as you can see in the left picture, there are 3 examples of different enzymes with different pH scales. As you can see, for pepsin is near, like, it seems to be 1.5. So how can I discern this? I think we are asking this often, that how can I determine the optimum pH of an enzyme in graph lines in a graph? So in this case, as you can see, the rate of reaction is increasing, and then it decreases immediately until the activity level is going to be 0. It is going to reach 0, as you can see in the graph for pepsin and all the enzymes. So these, let's say, so in this case, in my opinion, in my point of view, I'm going to provide this line which is being placed in the graph into 2 parts. The one which is going up and the one which is going down. So there is a point between these 2 connected lines, and that certain point is called the optimum pH, or let's say the optimum temperature of that specific enzyme, which in this case for pepsin, as you can see, it's nearly between 1 and 2, so 1.5. And for salivary amylase, as you can see, it's nearly 6, so we are going to say 6. And also for the third enzyme, let's say it's nearly 9. So we can determine also the optimum pH and the optimum temperature in a graph line. And also in the right picture, as you can see, it's related to the concept of temperature, which in this case, after the temperature is increasing, the activity level will increase too. That means they are proportional. But they are proportional until a certain point, which in that certain point, after that certain point, if the temperature rises from that certain and that specific point, immediately it goes down and the activity level will reach to 0. And this means that specific, let's say that certain enzyme has been completely denatured. So in this case, you are going to provide this graph line into 2 parts, the one going up and the one going down. The exact number, that specific point, that certain point, which has connected these 2 lines together in order to make a graph line, that certain point is called the optimum pH, or the optimum temperature of that specific enzyme. So that's all for today. Thank you so much. Don't forget to subscribe, like, and share. And if you haven't turned on the bell yet, please do it. So see you in our next video.